2020 was a great year for research in clinical microbiology. The COVID-19 pandemic presented unprecedented challenges, including challenges to clinical laboratories. Clinical microbiologists rapidly created a variety of tests for SARS-CoV-2, figured out innovative ways to collect and transport samples for SARS-CoV-2 testing, and also continued their research in areas outside of the pandemic. In this episode of the podcast, 3 editors of JCM are each going to discuss 3 of the most important papers published in the journal during 2020. Welcome to Editors in Conversation. This episode is brought to you by the Journal of Clinical Microbiology, available at jcm.asm.org and on Twitter at jclinmicro. I'm JCM Editor-in-Chief, Alex McAdam. This podcast is supported by the American Society for Microbiology, which publishes JCM. I'm joined by 2 experts to discuss the year in clinical microbiology, as seen in the papers published in JCM. First, Dr. Melissa Miller, who's director of both the Clinical Molecular Microbiology Laboratory and the Clinical Microbiology Laboratory at the University of North Carolina School of Medicine. Dr. Miller is also an editor of JCM and has been a guest on the podcast twice to discuss SARS-CoV-2 testing. You can follow Melissa on Twitter at MELMILLER-PhD, at Mel Miller PhD. Welcome, Melissa. I'm glad you could join us.
Thanks, glad to be back.
Also joining us is Dr. Patricia Simner. Dr. Simner is an associate professor of pathology at Johns Hopkins University and director of medical bacteriology. Trish has also been a guest on the podcast before to talk about detection of carbapentamases. You can find Trish on Twitter at SimnerLab, SIMNERLAB. Trish, thanks for coming back.
Thanks for having me.
Each of us has chosen 3 important papers or groups of papers that were published in JCM during 2020. We've agreed that at least one of the papers has to be about something other than SARS-CoV-2, and we're presenting them in no particular order. Trish, why don't we start with you? What's your first paper? Why did you choose it? What is it about?
Sure. So this is my token COVID-19 related article, and it highlights the impact the pandemic had on other areas of the lab and helped laboratories plan and address blood cultures as the pandemic progressed across the US. And so the title of the article is Bacteremia and Blood Culture Utilization During COVID-19 Surge in New York City. Sepulveda and others published this in JCM in August 2020. And so, as we all know, New York City was one of the first to be hit hard by COVID-19 pandemic in the US, which unfortunately progressed across the US from there. At that time, they were experiencing a surge in febrile patients with COVID-19 that led to a sharp increase in blood culture utilization, which overwhelmed New York City hospitals' capacity of blood culture instruments, among other resources, as we are all familiar with. So at that time, the data were lacking on the value and utility of blood culture in patients who are suspected or confirmed COVID-19 patients, many of who were treated empirically for potential bacterial co-infections. And so this was a multi-center study from 5 New York Presbyterian hospitals, where they collated their data from their blood culture orders from January 1st, 2020 to March 31st, 2020, and compared that data to a baseline during the same time period in 2019. And during that time, there were 88,000 blood cultures that were obtained from 28,000 patients. And what they experienced was a drastic rise in blood culture ordering in the second half of March, a 34% rise, with an associated decreasing positivity rate. So they decided to stratify the data further based off of whether the patients were tested for SARS-CoV-2 or not. And so based off of testing, blood cultures ordered from patients with SARS coronavirus 2 positivity had a positivity rate of 3.8%, compared to those that were negative for SARS coronavirus 2, which is 8%. And those that were not tested was a third group that they compared to with a 7.1% positivity for blood culture diagnostics. In diving in further into those few positives with the SARS coronavirus patients, there was about half of the positivity rate was due to contaminants. And so when those contaminants were removed, there was only a positivity rate of about 1.6%. And so overall, in comparing that time frame to the baseline, there was less than a third, the bacteremia rates were less than a third compared to baseline. And so what this study really showed is that blood culture diagnostics, the diagnostic yield of blood cultures is remarkably low for patients who are diagnosed with COVID-19. And so it really justified judicious use of blood cultures in patients who were suspected or confirmed with COVID-19. And this served as evidence to hospitals who were yet to experience the surge, that blood cultures in patients who were suspected of COVID or patients who were initially presenting, that the initial admission to blood order sets or specific COVID sets or guidance provided to physicians shouldn't include blood culture diagnostics unless there's a high clinical suspicion. So this allowed not only for labs and hospitals to prepare for this as the surge spread across the US, but also allowed for labs to prepare for the potential for them to experience the same thing, so to order up on their blood culture bottles, or as well as prepare for potential mitigation strategies, as the publication also discussed. Was they ended up implementing a 4-day incubation period over the standard 5-day incubation period to free up capacity on their automated instruments. I reached out to Dr. Westblade and Green, who are co-authors on this paper, to see if they still have adopted the 4-day incubation over 5 days, but they said they have returned to the 5-day incubation period. And I was curious because there was a recent publication in JCM entitled Evaluation of Optimal Blood Culture Incubation Time to Maximize Clinically Relevant Results from a Contemporary Blood Culture Instrument and Media System from Drs. Ransom and from Dr. Carrie-Ann Burnham's group, who they had evaluated the blood culture BACTEC alert virtual blood culture system and media, and found that implementation of a 4-day incubation period could enhance clinically relevant results by reducing recovery of contaminants and finalizing blood cultures 1 day earlier. So I feel like these two studies, although one was a mitigation strategy in terms of dealing with the pandemic to reduce the incubation period to 4 days, that with this newer study, this might be a way of the future that we might not need the standard 5 days as part of our blood culture incubation.
I like how you managed to transition from SARS-CoV-2 over to blood cultures.
I practiced that.
I remember when that first paper came into JCM, I remember thinking to myself, well, that was a smart thing to look at. That was kind of a creative thing to look at. While the rest of us are all pounding on various SARS-CoV-2 tests, they kind of went off on a different track and came up with something very interesting.
Right. I think it was very helpful to other labs across the US as they started to experience their own surge and try to determine whether blood cultures were useful, at least on initial admission. So it was helpful.
Yeah. Melissa, let's turn to you next. What's your first paper?
Well, keeping with the SARS theme, this is the only SARS paper I'm going to talk about. And there were certainly a large number of high caliber papers in JCM in 2020 related to SARS-CoV-2. And it was kind of hard to pick, but I had the privilege to read many of them as one of the virology editors. One topic that immediately came to my mind in terms of novelty and impact in our field was the role of saliva in diagnosing COVID-19. So hopefully now your mouth is watering. I'm talking about saliva. Mine always waters when I start reading these papers. And there's several papers that I could have chosen on this topic, but the one I settled on was in the September 2020 issue of JCM by Nagura Ikeda et al., entitled Clinical Evaluation of Self-Collected Saliva by Quantitative Reverse Transcription PCR, direct RT-PCR, reverse transcription, LAMP, and rapid antigen test to diagnose COVID-19. So you can tell from the title, it's a very comprehensive evaluation of the role of saliva. There's been a lot of discussion in the literature and in mainstream media and even on previous JCM podcasts about the need for highly accessible testing. And one critical aspect of this is having an accessible specimen type, ideally one which would be self-collected. So as clinical microbiologists, we've not really been accustomed to considering saliva as a specimen type to diagnose a respiratory viral illness. And I would venture to say many of us scoffed at this idea early in the pandemic. But between the shortage of swabs and the reluctance of people to routinely and repeatedly get nasopharyngeal, even nasal and throat swabs, it was clear we needed to think about other alternatives if the performance data allowed us to. So prior to this publication that I chose, the published sensitivity was really kind of all over the board for saliva, and it is still a bit. for SARS-CoV-2 RNA detection. And it ranged anywhere from 69 to 100% in some studies. Even in some of our own labs, we didn't publish our data, but we discussed among ourselves it was even lower than those that were published. So in these studies, there are differences among the types of patients testing, the sampling of the saliva, and even timing of collection that impacted the data in terms of sensitivity. In the paper by Nagura, Akita et al., they compared the performance of saliva in COVID-positive patients in 7 different tests. So there was a reference quantitative laboratory developed test that was endorsed in Japan, the Roche Cobas test, which is used widely and has EUA here in the US, 3 direct RT-PCRs using different pre-analytical processes to eliminate the extraction step, and then an RT lamp-based isothermal amplification, and then even a rapid antigen test. So when looking at these tests, sensitivities ranged widely, with the reference quantitative LDT RT-PCR and the Cobas test being the highest, with sensitivities around 81%. The direct non-extracted RT-PCRs and LAMP had sensitivities ranging from the 50s to 70%. And maybe not surprising to many of us, the rapid antigen test, the use of saliva in a rapid antigen test was 12%. So we know a lot about viral load now in terms of how it impacts sensitivity of tests. And so not surprisingly, the sensitivity was correlated with CT value of the specimen. Also time since symptom onset and whether the patient was symptomatic or asymptomatic. Persons who were symptomatic for less than 9 days had the best sensitivity for saliva. So interestingly, in this paper, there were no sophisticated instructions for saliva collection. It was basically spit at least 500 microliters in this tube. That was kind of the approach that we used too, knowing people don't follow directions. So there's another JCM paper by Prokop et al. In the November 2020 issue, and it shows that an enhanced saliva collection, which it's hard not to act this out. I promise I won't act it out.
Go for it. Do it.
We describe this as a strong sniff, elicited cough, and then collection of that saliva or those secretions. And when they compared that to NP swabs, they had a 100% positive percent agreement. So I still think there's room for improvement for how the saliva specimen is collected to improve sensitivities. But this paper that I showed did a really thorough job in showing how it performed different. The same specimen performed differently in different assays.
I think this paper is kind of illustrative of the explosion of creativity that there was early on during the pandemic, with people looking for alternative specimen types and then looking for alternative methods for collecting, nasopharyngeal specimens. For example, I think we had a nice paper about 3D printed swabs from Dr. Arnaud at the BIDMC, for example. Are either of you using saliva in your laboratories to test for SARS-CoV-2?
We validated it, so we were sitting on ready, but we never pulled the trigger and implemented it.
How about you, Trish?
Just a recent saliva test that went live for our healthcare workers. And so it was just to have access for them. It's not directly in the clinical lab here. It was part of our genomics center who took on that testing for us. But there is now a saliva test that's live. So.
Gotcha. We started a validation of it, and the positivity rate at the time was pretty low. It's much higher now than it was then. And we were having a lot of difficulty recruiting patients to give us samples for it. So we have tabled it. And as the swab supply has loosened up, it seems to have been okay for us.
It's interesting because I heard that some people are having troubles getting the saliva, producing the saliva, and sometimes it's taking them just 25 minutes to be able to produce the amount that's required of them. So
we actually did a time study when we were doing self-collected salivas for our validation. And on average, it took us 3 minutes to collect enough saliva to validate all of our tests. So
it's a lot longer than I thought. Yeah. I think that's the reason we couldn't recruit people. I think it's because the staff that was collecting samples really has to keep things moving.
Right.
And it was gumming up the works to have patients sitting in their cars spitting into tubes.
Right.
Yeah. Well, I'm going to jump to my first paper, which is about SARS-CoV-2, and it is neutralizing antibodies correlate with protection from SARS-CoV-2 in humans during a fishery vessel outbreak with a high attack rate. The corresponding author on this was Alexander Greninger at the University of Washington. This is a paper that came into JCM in August when there was very little known about the immune protection against SARS-CoV-2. And it was the first demonstration that the presence of neutralizing antibodies against the virus correlates with non-susceptibility to infection by SARS-CoV-2. I was really pleased that we got this in JCM. This is sort of an interesting outbreak that occurred again on a fishery vessel. And for reasons that are not clear in the paper, all the people on the vessel, or nearly all of them, had been tested within 2 days of departure of the vessel. So out of 122 people on the vessel, 120 were tested by reverse transcription PCR for SARS-CoV-2, and they also had samples collected for an Abbott test for antibodies to the nucleoprotein of SARS-CoV-2. And the authors point out that the high population density and the large amount of contact contributes to high attack rates of SARS-CoV-2 on vessels. So before the ship left, everyone who was tested was PCR negative, and 6 people had positive tests in the antibody test. And clearly the PCR had missed something because there was an outbreak on the ship while it was out to sea. Subsequent testing, I'm going to back up a little bit. I said that 6 people had positive antibody tests. Subsequent testing demonstrated that of those 6 people with the positive antibody tests, 3 of them actually had neutralizing antibodies, that is, antibodies that could prevent SARS-CoV-2 from infecting cells. And they determined that using 4 different tests, probably most importantly using pseudovirus neutralization with a lentivirus that had the SARS-CoV-2 spike protein on the surface. So 3 people had neutralizing antibodies before the vessel left. The vessel returned to port 18 days after it had departed, and that was because there was a case of COVID-19 in a crew member who required hospital admission. Upon return, everyone was tested, not everyone, but nearly everyone was tested repeatedly by RT-PCR and serology for up to 50 days after the time of return of the vessel. 101 of the people tested positive by reverse transcription PCR with a CT value less than 35, which was their defined definition of a positive result. And that happened within 10 days of return of the vessel. Nearly all those people converted in the Abbott assay as well. There were 3 additional crew members who seroconverted upon return who did not have positive PCRs. 2 of them had weekly positive PCRs, but didn't meet the definition of positivity. So the important thing here was that the 3 people who had had pre-existing neutralizing antibodies did not become infected, despite that very high attack rate in the population on the vessel. So that was as determined both by symptoms and repeated RT-PCR testing of those folks. And the p-value, when you look at the whether that is a significant observation, was 0.002. So it was certainly not by chance that those people did not become infected. So my takeaway from this was that this sort of interesting natural history experiment gave us really the first evidence that antibodies could provide protection against infection with SARS-CoV-2. And that came at just the right time because it really lent strong support to vaccine development for SARS-CoV-2. I'll stop there on that one. And then Trish, why don't we go back to you? What's
Sure.
your next paper?
So I have a set of papers for this one. I'm going to discuss artificial intelligence combined with digital slide scanning for assessing trichrome-stains for intestinal protozoa, as well as for assessing gram stains to assign Nugent scores for bacterial vaginosis diagnosis. And so we know that manual light microscopy of stools for intestinal protozoa by Trichrome-Stained and gram stain for Nugent score and the diagnosis of bacterial vaginosis still remains the gold standard, despite there being the introduction of molecular tools. Yet it's still a labor-intensive, time-consuming, and requires considerable expertise. And so in these 2 studies, they apply machine learning and digital slide scanning that shows the promise to revolutionize the way we analyze slide-based techniques in the clinical microbiology lab. In these 2 studies, they use this mathematical convolutional neural network to assign slides as either being positive or negative. And I can envision the use of digital slide readers combined with artificial intelligence tools as a component of total lab automation in the future. And so the first study is entitled Detection of Intestinal Protozoa in Trichrome-Stained Stool Specimens by use of Deep Convolutional Neural Network. And this was published by Blaine Matheson and Mark Couturier in JCM in June 2020. And so the goal of the study was to create a sensitive model using a deep learning-based convolutional neural model that could screen out negative trichrome slides while flagging potential parasites for manual assessment. And so what you have to do with these artificial intelligent models is that you need to first train them. And so they trained them by assigning classes to the protozoa. And the classes were targeted species and morphologic stages. And so, for example, for Giardia duodenalis, they had a class for the cysts, and then also a Giardia duodenalis class for the trophozoites. And for each class, they had anywhere from 1,000 to 20,000 exemplars per class used to train the model. I could only imagine the amount of work that took just to find the slides. It's, in thinking through this study, it's fascinating how much time and effort went into this study. So after they trained the model, they then wanted to validate it for clinical use. And they did this by using 10 unique positive slides per class and 125 negative slides. And overall, they found a percent positive agreement of 99% and a negative percent agreement of 98%, despite them, going more for high sensitivity than specificity. And they found really excellent reproducibility. And what I found the most fascinating was that their limited detection studies using serially diluted stool was five times more sensitive by using the artificial intelligence method than by manual exam by multiple parasitologists. And so that's an exciting study. I followed up with both Blaine and Mark, and they're using this clinically, and they're further validating additional protozoa to include into their model as they move forward. So it's just really cool to see this method not just being developed, but actually being used clinically. And I could see this, being adopted more and more in the clinical lab in the future. And so that leads me into the second paper, which is Deep Neural Networks Offer Morphologic Classification and Diagnosis of Bacterial Vaginosis. This is published by Wang and others. That was posted online on November 4th. And they developed and optimized the same neural network model, the same idea or the type of model, and evaluated its ability to identify and classify the three categories of Nugent scores. So that being normal microbiota versus altered microbiota versus bacterial vaginosis, from microscopic images in comparison to the manual assessment. Like the first study, they had a training set using 23,000 slides to use to train the model, and then a validation set that was from 3 different hospitals where they obtained up to close to 6,000 samples to then validate the model that they had trained. And so simply looking at whether you could divide normal microbiota versus altered and bacterial vaginosis in 2 separate categories, kind of negative or positive. They have a sensitivity of 82.4% and a specificity of 96.6%. And compared to competent technologists reading these slides. And then they took it a step further. They said, okay, now we want to see how it would have done if we would have broken it up into the classifications. Now, not just into the 2 categories, we want to do normal microbiota, altered microbiota, and bacterial vaginosis. And so using that, they found that their model was 75.1% accurate in assigning to the 3 categories of Nugent score, which was 6.6% higher than the average of 3 technologists. So it did better than the average than the manual assessment, both for the accuracy as well as reproducibility in assigning the 3 categories of Nugent scoring. So again, 2 really interesting studies that took probably a ton of work to get to the finish line, but shows the potential of the applications of artificial intelligence and digital slide scanning that could be used in various areas of the lab. And like I said, I envision this being part of a total lab automation line in the future.
Melissa, what do you think? Artificial intelligence coming to the clinical microbiology laboratory?
Well, I'm sitting here thinking that there's both sides of the coin, right? Especially with such highly complex stains and slides like BV and parasitology. When we talked about the parasitology paper in our lab, it was like, bring it on. I mean, we spend so much time looking through negatives. Our positivity rates are very low. So if the negative predictive value is very high, this is very welcome. As soon as you start to kind of embrace that, you start seeing a little bit of resistance. And I was actually going to ask Trish, what she thought in terms of the impact to needing skilled technologists and the level of staffing that we need once we have artificial intelligence.
Yeah, I think we still need those skills. And so as demonstrated in the first paper, they made it so it was highly sensitive. So you're still getting a lot of junk that's popping up as images and stuff, but they wanted it to be more sensitive so the technologists could still spend their time criticizing those that could potentially be positive. And they made it so their algorithm is simply, we think there's something here of interest versus not. So the technologists still in the end are the ones doing the final diagnosis, still requiring that specialized training, just not needing to spend hours on end reading those negative slides. And we had to make a difficult decision not too long ago to send out our O&P because we were losing staffing and couldn't replace them. And O&P is just not the critical test that we need staffed. And so we eventually sent them out. But I think this is something that would have been definitely helpful to, help with staffing issues around this.
Yeah, I think that's a really important message.
So
I think so too. And I think artificial intelligence is going to be the next big wave. Once things settle down a little bit with SARS-CoV-2 and we can refocus, I think this is going to be a really important part of clinical microbiology. We had a nice paper from James Kirby about using artificial intelligence to interpret gram stains. And then Nate Ledebauer has had a couple of papers about using artificial intelligence to actually read plates, for urine cultures and that kind of thing. So I think this is coming. Let's see. I think, Melissa, we're over to you.
Okay, so my next two topics are both metagenomics, but a little bit different takes on them. So this is something that I have been particularly interested in myself in terms of the role of metagenomic next generation sequencing to diagnose infectious diseases and how best to use this powerful technology. You can read a point counterpoint in JCM about this, which was in the March 2020 issue. The first paper I'll discuss in this round is by Kingery et al. Was in the November 2020 issue. And the title is Targeted Metagenomics for Clinical Detection and Discovery of Bacterial Tick-Borne Pathogens. There's also an accompanying commentary in the same issue you may wish to read by an infectious disease specialist, Dr. Matthew Tyndall. To lump tick-borne diseases into one nice syndromic package is really misleading from a diagnostic perspective due to the large number of potential pathogens that could be represented in this group, as well as geographic differences in prevalence. Diagnosing tick-borne disease relies heavily on the combination of PCR and serologic tests, and physicians must order each test individually. And in doing so, risk missing an unexpected diagnosis or maybe a diagnosis that should have been expected. The syndromic panel that you might build in different areas of the country and certainly in different areas of the world would be very different based on geography and patient exposure. So in this paper, the authors use targeted metagenomics approach by first amplifying the variable region B1, B2 of the bacterial 16S ribosomal RNA gene prior to high throughput next generation sequencing so they could detect or assign taxonomic predictions of bacteria in blood, synovial fluid, spinal fluid, and tissue from patients suspected of having tick-borne illness. Most of the specimens were blood specimens. So this targeted approach has the advantage of enriching for bacterial sequences in a complex sample environment that's rich in host sequences. But keep in mind, it's limited to bacteria. It's not also detecting protozoa or viral causes of tick-borne diseases. So the numbers in this paper are impressive. They were specimens that were submitted either to Mayo or to Vanderbilt, over 1,000 control specimens and over 13,000 samples, most of which were blood. that were submitted for tick-borne disease diagnostics. So of these, the authors identified 881 positive samples that had taxonomic predictions for either Anaplasma, Borrelia, Ehrlichia, Francisella, or Rickettsia. Most cases were representing a single etiologic agent, but there were 3 specimens that had 2 potential pathogens. Again, something that we certainly see in tick-borne illnesses. Using secondary confirmatory methods, and these consisted of specific PCRs or additional sequencing, 99.5% of these were confirmed. Using PCR diagnostics alone, of those bacterial species that were diagnosed, 6 of them would be covered versus the 12 that were covered by Metagenomics. So an additional finding was the detection of organisms, at least taxonomic predictions of organisms in 5 specimens that were in the Rickettsia, but not known tick-borne human pathogens. So this is showing how this technology might be used in combination with diagnosis, but also for the discovery of new or previously not described causes of tick-borne illness. Another benefit of this agnostic approach in this study is they identified cases of Q fever and leptospirosis, and all but one was confirmed by secondary testing. So again, getting back to that, perhaps an unexpected diagnosis would be picked up from this method. Of course, normal clinical specimens and water amplify bacterial sequences, so this has to be taken into account with any diagnostic reporting algorithms. And I thought the authors did a really nice job of discussing that in this paper in terms of the types of sequences they detected and how they categorized those as things that wouldn't be reported for this particular need. So a benefit of applying kind of a targeted bacterial metagenomics approach to something like tick-borne infections is the ability to narrow the focus of the organism. So you can kind of get these environmental organisms or organisms you wouldn't expect in this type of diagnostic out of the reporting scheme, instead of reporting any and all bacterial sequences obtained, which is what we'll talk about in my next topic. So from a practical standpoint, right, providers that are trying to diagnose tick-borne disease could potentially have one orderable test, at least for the bacterial causes, with what was described in this paper. What's not known, this is talked a bit about in the commentary, is the true clinical sensitivity of this approach, right? So they're just looking at, specimens coming in which providers are considering tick-borne illness, but we don't know how many people truly had bacterial tick-borne illness that were negative by targeted metagenomics. So I still think it's really exciting to see how to thoughtfully apply such a powerful technology to a diagnostic dilemma that we all struggle with.
Great.
Thank you. Yeah. Sorry, Trish, please go ahead.
Do the authors have any intent, or do they plan to, further multiplex their targeted panel to try to not just detect bacterial? Is there?
That wasn't mentioned in the discussion, and I didn't have direct conversation with the authors to see what their next steps were regarding that. But this was certainly a focus on bacterial causes.
Very cool.
It's a great one. Yeah, I really like that paper. Let's see. I'm going to jump in with my next one. And I've actually got a group of 4 papers. I'm not going to read the titles for 04 because it will take all the time I've got for this, but I'll focus on one of the papers. And the reason I chose these 4 papers is because they're good papers and they are important, but they also illustrate something that happened due to the SARS-CoV-2 pandemic that has never happened in clinical microbiology before. And that's sort of the tremendous attention that came to clinical microbiology as a result of the pandemic. So the paper I'll discuss in detail is performance of Abbott IDnow COVID-19 rapid nucleic acid amplification test using nasopharyngeal swabs transported in viral transport media and dry nasal swabs in a New York City academic institution. The corresponding author on this was Maria Aguero Rosenfeld at NYU Grossman School of Medicine. And I'll put links up to the other 3 papers that looked at the same assay with the show notes. And I'll mention those in just a minute as well. But the significance of these papers comes in part on the reliance of the Trump administration on testing to prevent transmission of SARS-CoV-2 within the White House. And I'll come back to that at the end. As I said, I'm going to focus on just one of these papers, And it's a relatively short paper, very straightforward. In the first part of this, they were comparing the IDnow assay from Abbott to the Cepheid Expert Express. Expert Express is a conventional reverse transcription PCR, albeit one with a relatively rapid turnaround time of 45 minutes. And there are a number of papers showing that it's comparable in sensitivity to other RT-PCRs. In the initial comparison in the paper I'm talking about, they used sample excess or leftover specimens that had been tested in the Cepheid assay. They had 2 of them, and they did a serial dilution of each. One of those samples had a high viral load, one had a low viral load, as determined again on the Cepheid platform. The IDnow assay worked well with the sample that had high levels of SARS-CoV-2, but it was less sensitive when the sample with lower viral load was tested. They then described very briefly testing a series of previously positive samples and they found the same thing. The lower the level of virus as determined in the Cepheid assay, the lower the sensitivity of the Abbott IDnow. One of the concerns in this initial part of the study, which they then address, is the sample type. And in a number of these papers, primarily for convenience, authors used leftover nasopharyngeal swab material in viral transport media or universal transport media, but the assay is recommended to be used with dry swabs, and they did that in this paper. So they next tested dry nasal swabs, which are the recommended specimen type for IDnow, as I said, and compared that to nasopharyngeal swabs in viral transport media using the Cepheid assay. So they collected both specimen types. They tested 101 samples, 31 of which were positive in the Cepheid test, which was the reference test. The IDnow detected 17 of those 31 positives for a positive percent agreement or sensitivity of 54.8%. Of the 70 specimens which were negative in the Cepheid assay, 69 were negative in the IDnow, so the negative percent agreement was very high at 98.6%. So that's the one I'll talk about in detail. I just want to mention a few others, one by Amanda Harrington, in which she looked at she and her collaborators looked at 524 samples and found a positive percent agreement of 74.7% using the Abbott M2000 RT-PCR test as the reference standard for the IDnow. There's a nice study by Dr. Berry in which they looked at 108 samples and found a positive percent agreement of 87.9% using a composite reference standard of 3 different tests, including the Expert Express. And finally, Dr. Rhodes looked at 96 samples previously positive in a modified version of the CDC assay. And in that study, they found a somewhat higher positive percent agreement for the Abbott IDnow of 94%. And I don't have a good explanation for that. The others found somewhat lower positive percent agreement. And the 3 studies I just discussed did have samples that were collected and placed in transport media. So that's an important caveat to keep in mind. My takeaways from this, I think it's fairly clear that the Abbott IDnow is somewhat less sensitive than other nucleic acid amplified tests, and people should keep that in mind when deciding whether to use this test. But more broadly, thinking about this, I think most, maybe all of us, would agree that testing alone, no matter how frequently done, cannot adequately prevent transmission of SARS-CoV-2. But if you are going to rely on testing as a mainstay to prevent transmission, using a very sensitive test is probably a good idea. And the former head of the FDA, Dr. Scott Gottlieb, has said that the Abbott test was preferred over other tests because of the very rapid turnaround time. And that turnaround time is about 8 to 13 minutes. In my opinion, the use of the Abbott IDnow may well have contributed to the transmission of SARS-CoV-2 in those who were working in the White House during the Trump administration. Any thoughts, questions?
I think you hit the nail on the head, right? It's not testing alone, right? I mean, there's been so much focus on testing, which is, of course, a very important part of the equation, both diagnostics and transmission. But we can't look at testing in a silo. We need all the other W's that you mentioned to continue to prevent transmission. And we're at our highest rate right now in North Carolina, which, in the other podcast, if you would have told me in January 2021 would be the worst, I probably would have quit right then and there. But, here we are, so desperate times call for desperate measures. So some of us have to use less sensitive tests to expand our breadth, throughout our state to get to rural areas, and that's okay. But I think just not doing it in a silo.
Yeah, and I think we have to understand the performance of these tests. The antigen tests are the other group where there's somewhat less sensitive, but that doesn't mean that they can't be used appropriately. They just have to be used with an appropriate understanding and reflex testing where that's needed.
Great.
Trish.
Okay, my
what's
last one.
Your third, your final paper.
So I was very excited when I was looking through the potential manuscripts to choose from in seeing a whole lot of nanopore sequencing manuscripts that had been published in JCM. So nanopore sequencing is a long-read third-generation sequencing platform, and it can come in a portable sequencing platform. And the real potential to this in the clinical lab is that it allows for real-time base calling and analysis of the data in real time. And so I've always been excited about the potential of this technology, but it's mostly been, most of the publications have been in the research domain. So I'm super excited to now see a whole slew of papers on nanopore sequencing, different applications in JCM, meaning that we are now validating them to a point to apply them in the clinical care. And so the paper I chose was Metagenomic Nanopore Sequencing of Influenza Virus Direct from Clinical Specimens, Respiratory Specimens. And so they applied metagenomic next-gen sequencing from throat and nose swabs. And so metagenomic next-gen sequencing means that you're extracting all nucleic acid from the sample and you're sequencing in parallel. This is not a targeted approach. You're not choosing which nucleic acid from that sample that you're actually going to be sequencing. Everything in there is going to be sequenced, including host. And so they applied a cDNA approach because they're looking for influenza virus, obviously. And they used this randomly amplified sequence-independent single primer amplification, or CISPA approach, to randomly amplify the nucleic acid to be able to detect recovery of lower abundance genetic sequences and samples prior to sequencing on nanopore platforms. And they multiplexed up to 12 samples on a MinION or a GridION. And so they compared metagenomic next-gen sequencing, this technique, compared to GeneXpert of 40 known positive influenza A or B specimens that were positive by the GeneXpert system and 10 negatives. And they found overall the percent positive agreement was 83% and the negative agreement was 100%. But the positivity rate or the percent agreement really was dependent on the viral titers. And so they divided the samples that were positive into those of mid to high viral titers based off of the crossing or cycle threshold. So anything greater or less than 30 was considered mid to high viral titers. And they had 27 of those samples, and they were able to detect all 27 of those using the metagenomics approach. And then they had 13 low viral titers, which were those with a cycle threshold of 30 to 40. And so I think it's got pretty good agreement with standard of care methods. They also did a limited detection study where they were able to detect 10 to the 3 genomes copies per ml in pooled samples, showing a strong correlation between viral titers and the proportion of influenza reads that they were sequencing. Now, the power of metagenomics in this case comes in the ability to potentially get coverage of the entire viral genome. And so in this study, they looked at coverage of viral genome and found that it was impacted, not surprisingly, by your host and your bacterial reads if there was in high abundance in that sample, but also by the titers of the viral pathogen, by influenza titers itself. And they were able to reconstruct greater than 99% complete sequences for all 8 gene segments for influenza in those samples that were considered high viral titer. And so the ability to be able to kind of, let's say, whole genome sequence through a metagenomic approach allows us to, study molecular epidemiology, transmission events, the potential public health interventions, and estimate vaccine efficacy, and even look at drug resistance mutations in this diagnostic that can tell you, whether the patient's positive or not, but you can get a whole lot of additional information if you have sufficient coverage of the viral genome. In addition to being able to provide that information, you can also detect co-infections. In this case, they did a cDNA approach, so they were looking for RNA pathogens. They also did a DNase treatment, so it was specific to viral pathogens, where they were in one case able to detect a co-infection with human coronavirus, not SARS-CoV-2 co-infection. And in one of the negative samples, they were also able to detect something that was unknown before. So it just shows the potential of this technology of not just presence or absence of a particular pathogen, but also the additional information that metagenomics can yield. So the authors concluded that further optimization was required to improve sensitivity to get to a point where they would want to see this in the clinical setting. But the study does show the promise for the Nanopore platform to be used in the diagnosis and genetic analysis of influenza virus. And as I mentioned, there was a whole slew of different papers on nanopore sequencing, and anywhere from the bacteriology to the fungal and the mycobacteriology side, it was covered in the different papers that were presented. There was a sample to report solution for taxonomic identification of cultured bacteria in the clinical setting, using nanopore-based sequencing by Neuenschwander in June of 2020. Nanopore sequencing of the fungal intergenic spacer sequence as a potential rapid diagnostic assay by Morrison and others in December 2020, and then application of targeted next-gen sequencing assay on a portable sequencing platform for culture-free detection of drug-resistant tuberculosis from clinical samples by Kabibi and others in October 2020. So just showing the diversity of papers that were submitted and published in JCM this year on nanopore sequencing. In addition to those papers, there was commentaries as well as some very nice mini reviews on the topic. So I was just excited to see this because I've always seen the potential of the sequencing platform. So I'm excited to see it almost close to the clinical care setting now.
It's been a lot of energy, yeah.
As these have become more and more portable, what's been happening to the cost? Because that's obviously a big consideration to making this routine in clinical laboratories and less of a research or epidemiologic tool.
That's a great question. So they're now working on a flongle, which is an adapter that you could put in there. It's less throughput than your standard MinION flow cell. But it allows for the cost to come down. For that flow cell, I think it's around less than $100 right now. And then the other fun thing about this technology is that they've, made some of the library preparation techniques a lot more simple and rapid. And so you could really go from sample to on your sequencer within 4 hours, even less probably at this point. And then depending on how much sequencing depth you want, you could stop your sequencing run at any moment to stop and say, okay, I got enough sequencing data to assess my samples. So, within a workday, you're getting your results, if not more rapidly. So cost effective and more rapid are where they're going right now. So
awesome, thank you. Melissa, am I right that your last paper is on metagenomics as well?
It is. I'm going to stay on the sequencing train, and I've picked two papers, and I'll just discuss each one briefly, and you can read them for more details. So this really gets to the clinical utility. So less the implementation of or the sensitivity and specificity, a bit about that, but how are these being used clinically? So unlike the tick-borne disease paper I talked about earlier, we're not talking about a targeted approach. As Dr. Simner just said, we're looking at shotgun metagenomic sequencing, anything that's there. So you can identify sequences of organisms across taxa, even kingdoms. So the first paper is by Rodino et al. From Mayo Clinic. Robin Patel was the corresponding author in the December 2020 JCM issue. Retrospective review of clinical utility of shotgun metagenomic sequencing testing in cerebral spinal fluid from a U.S. tertiary care medical center. The second paper is from Niles and colleagues from Texas Children's Hospital, where Paula Ravel is the corresponding author, and this is in the November 2020 issue. So I guess I was liking the late-breaking papers of 2020. Maybe I finally came out of my COVID fog and read things about COVID by October, November. And so this one is Plasma Metagenomic Next Generation Sequencing Assay for Identifying Pathogens, a retrospective review of test utilization in a large children's hospital. So Rodina et al. Looked retrospectively over 2 years at the results obtained on spinal fluid sent to the University of California, San Francisco for metagenomic sequencing. As many of you know, UCSF offers a reference laboratory service for this, and they've previously published their work in New England Journal of Medicine in 2019. Most of us have been involved in discussions either with infectious diseases or neurology about patients for whom there's no diagnosis for their central nervous system disease, and often an infectious etiology is suspected. And we know that up to 50% of patients with encephalitis never receive a definitive diagnosis in terms of the cause. So these challenges really open the door to applying sophisticated technologies like metagenomic sequencing to try to diagnose patients that may have an infectious cause. But there's really little detail on the clinical utility or the yield of this approach. So over 2 years, Mayo had sent 80 specimens to UCSF. 12 of these were positive. Of these that were positive, 7 out of 12 were considered false positives or likely contaminants. So that left 5 that were positive with potential pathogens, which were Toxoplasma, West Nile, Abunya virus, HHV6, and an enterovirus, D68. The authors also comment on the utility of negative results because that also can be very informative, again, if the negative predictive value is very high. And about 15% of the negative cases, the reporting had comments regarding potential decreased sensitivity. And for example, this could be due to high background nucleic acids present, as Trish just mentioned as well. And so that brings into question kind of the validity of some of the negative results. And so the authors discuss the rate of clinically impactful results was low based upon their experience of the specimen sent. And then when you start looking at the cost and the cost per impactful result, this becomes quite expensive. And so again, there's more to be done in this area in terms of finding exactly. And in both of these papers, there was no restriction on who got to send the test. And there have been other papers that have been published. Do you look at cell count? Do you look at narrow in on who should be tested? And the second paper from Paula Ravel's group at Texas Children, they looked at plasma metagenomic sequencing sent to Carius, which is a commercial lab offering this service. So in this test, they're looking for cell-free DNA circulating in the plasma as a surrogate for a variety of infectious syndromes, which some of which have been described as pneumonia and sepsis, also meningitis and others. The interesting part to me about this paper is that the metagenomic testing was sent concurrently with the conventional testing. So they could really assess whether the cell-free DNA metagenomics offered an increased diagnostic yield and improved time to detection, as has been suggested in some other publications. So spoiler alert, it didn't. They sent 60 patient samples over 3 months. So they're sending a lot more kind of on a monthly basis in their pediatric population, at least for the study. Most of the kids were immunocompromised in some manner. And the authors report a 61% positive agreement, 58% negative agreement between metagenomics, the cell-free DNA metagenomics, and conventional testing. In terms of time to results, the conventional testing was reported about 3.5 days sooner than the metagenomics, probably mostly due to shipping and packaging and having to send it out, but still the results were available sooner. In cases where metagenomics identified organisms that weren't found by conventional testing, and their conventional testing was, culture, PCR, serology, histopathology, and even some broad range sequencing. So in these where metagenomics identified something that conventional didn't, a change in therapy only occurred about a quarter of the time. There were 3 pretty significant clinical cases that were missed by metagenomics. One was a TB case, HSV1, and a Staph aureus case. Again, supporting that this technology, at least at this point, is an additive diagnostic strategy. It is not a replacement for conventional tests. So as with the previous paper I mentioned from the Mayo group, these clinical utility data are really important to get into the literature. I'm really proud to have them in JCM as we try to determine, what's the yield? And sometimes the answer is, well, it wasn't high yield in this patient population. So then we can go back and start determining where this technology might have its greatest clinical impact without breaking the bank. Obviously, there's more work to be done in this area, and we would love to see your papers on this. So,
yeah. Are you, each of you, is your lab or your institution sending samples to Carius, sending samples to UCSF?
So
we do not routinely.
How about you, Trish?
So we validated metagenomic next-gen sequencing from CSF, and so we now offer that in-house, but with a whole lot of caveats around when it can be ordered. And so I do think diagnostic stewardship is extremely important, as Melissa highlighted, and these studies point out with these expensive tests. They're not always the right answer to help with the diagnosis. And so, and then we've sent out very few to Carius, and it's studies like these that help me in discussing with infectious diseases or other clinical teams why, we should wait until we get some of our standard of care results back, or why it might not be potentially helpful, or especially with the cell-free DNA from plasma, they see a lot of, just asymptomatic individuals having bacteria floating around. And so, that positive result, it's scary to me to think that, the teams might act on something that is really just junk, or, I just really think there needs to be strong diagnostic stewardship and a real precision medicine team that works around the hospital sending these out until we have further data to help us support when these are truly the right place to be using this diagnostic.
We send them, we require that either myself or my colleague, Dr. Pollack, the other clinical microbiologist here, approve it in each case. So they have to come through us, and typically we'll approve it if it's infectious diseases after some discussion with them. They're very level-headed about ordering it here. And I should mention, Dr. Pollack also published a similar paper to the one by Dr. Ravel, talking about our experience with Carius testing, and that's in JCM as well. Try to remember to put that in the notes. I'm going to do my last paper. I'm going to try to do it quickly if I can. And it is my non-Carius paper. And the title was Reduced In Vitro Susceptibility of Streptococcus pyogenes to Beta-lactam Antibiotics Associated with Mutations in the PBP2X Gene is Geographically Widespread. And James Musser at Houston Methodist Hospital was the corresponding author on this. Just by way of background, I think most listeners or all listeners will know that there have been no isolates of Streptococcus pyogenes found with true resistance to penicillin or other beta-lactam antibiotics. But there was a previous paper in Clinical Infectious Diseases by Dr. Venice in which they had found two isolates of Streptococcus pyogenes that had mutations in the gene for PBP2X, and those had elevated MICs with ampicillin, amoxicillin, and cefotaxime. PBP2X is a transpeptidase that synthesizes peptidoglycan, of course, and it's a target of beta-lactam antibiotics in group A Streptococcus. Mutations in PBP2X could lead to increased MICs with beta-lactam antibiotics in that species. So in Dr. Musser's paper, what they did was to look at genome sequences they had already determined in the past. So they had sequenced 7,025 group A Streptococci in three different groups, EMM groups, EMM1, 28, and 89. EMM is the gene that encodes M protein, which is a virulence factor for group A Streptococcus, of course. But in this case, they were using the sequence of the hypervariable region of EMM as a typing method for group A Streptococcus, and it's a common typing method for that organism. Most of the isolates in this collection were from invasive infections, including things like necrotizing fasciitis or bacteremia. They analyzed the sequences for mutations in PBP2X, and isolates that had non-synonymous mutations in PBP2X were tested for susceptibility to beta-lactam antibiotics. And non-synonymous means, of course, that it changes the amino acid sequence. And to start with what they found in the genotypes of those isolates, those 7,000 plus isolates, 137 of them had a total of 37 non-synonymous mutations in 36 different codons in PBP2X. They also found several synonymous mutations, but the non-synonymous mutations were overrepresented, and that's what you would expect to see if positive selection were occurring. In other words, if selective pressure in the form of, for example, antibiotics were selecting organisms. They found 4 non-synonymous mutations in multiple isolates of EMM 28 and 89 types, and that suggests convergent evolution of those different genetic lineages due again to that selective pressure. Isolates that had the same mutations and the same EMM types turned out to be closely related to each other, suggesting that they had spread in the community and were derived from common ancestors. So that's the genetic piece of it. And then when they looked at susceptibility, they also found some interesting things. They screened all the isolates that had mutations in PBP2X for growth with 6 nanograms per milliliter of penicillin in a solid agar or 15 nanograms per milliliter of ampicillin. And just to put that in context, those are very low levels of antibiotics. Those are 16 to 20 times lower than those that are used to define group A streptococcus as susceptible to those antibiotics. But nevertheless, those are levels that are high enough to prevent wild-type group A streptococcus from growing. And they found that several of the strains that had mutations in PBP2X were able to grow on those screening agars, but several of the strains did not. And then they used gradient strips to determine the MICs to several different betalactam antibiotics. And here again, they found that MICs to some of those antibiotics were elevated, and it varied between strain to strain which ones specifically had gone up. And then finally, isolates with one specific mutation at amino acid 601 in PBP2X had the highest MICs with all of the beta-lactams that they tested except for cefoxitin. And when they replaced the mutant with the wild type gene, the MICs fell significantly. And that was a nice direct demonstration of the importance of the mutation in elevating those MICs. I'm going to skip past some interesting structural work, and you can take a look at the paper if you're interested in that. But my takeaways from this were that the clones of group A streptococcus with mutations that elevate MICs to beta-lactams are relatively widespread. It's a nice follow-up to this paper using isolates from Iceland as well that people can take a look at. And this is something that bears watching. Again, this is not resistance. These are small elevations in the MICs, but I think we need to keep a close eye on this to see whether those whether the accumulation of additional mutations will lead to true resistance in group A streptococcus. So I think that's it for our papers. Do you want to do a quick game?
Sure.
What do you think?
Sure.
All right. This is a game. I got the idea from this from a column by Emma Goldberg in the New York Times. And she talked about the new words or the phrases that have come into use or become popular during the COVID-19 pandemic. So I'll give you the definition, and the two of you will work together to try to come up with the word. Some of these are pretty easy and some are pretty common. A few of them are a little bit harder. So
I can't speak most of the words I came up with during the pandemic.
These are all appropriate for all right. So our first one is skin outbreaks that are caused by the use of masks.
Maskne.
Yep.
Maskne. Yes. I have an adolescent son who has a little bit of a case of maskne. All right. Next one. This is another one that's pretty straightforward. When you spend a lot of time looking at all the bad news on social media.
Doom scrolling.
Yeah, doom
I
scrolling.
was bringing that
Together, Trish. Sorry, I should consult with you.
No, that's okay. You did good. I didn't know that one.
I am very good at doom scrolling.
We got to stay away from it. I also found doom surfing, but I heard doom scrolling a lot more than that. All right, the next one is one that I suffered from during the first podcast, and that is a hairstyle resulting from the inability to get a haircut.
I don't know either.
Take a guess.
COVID hair? I don't know.
Yeah, so a lot of these are just, Corona or COVID tacked onto something. So this one was Corona cut.
Oh, nice.
All right, the next one is an alcoholic drink that one has alone because of isolation to prevent transmission of disease.
Quarantini.
Quarantini.
Yes, nice.
Coronita is another one I found. Yeah. All right. The next one is the weight that a person may gain due to spending time in isolation.
COVID-19.
COVID-19. COVID with any number. COVID-10,
Yes.
COVID-11. All right. Next, something that we have all suffered from, and that is the exhaustion of too many online meetings.
I don't know, Trish, maybe like Zoom fatigue or something like that. Is there a fancy word for that?
Webinar fatigue. I don't know.
Zoom fatigue is what I had for that.
Okay.
That's a common one too. All right. This is one I hadn't heard before. A region with an increased real estate value due to people working remotely.
A region with an
A
increase.
city or a town.
Oh, you want a name of a city or a town?
No, I want to name 4 such cities or towns.
Oh, okay. Oh,
I like this one.
I don't know.
COVID, I don't know.
It's not a boom town. It's a
Zoom town.
Zoom town.
Yes.
Zoom town is right. I love that one. All right. Who are the people that you see during isolation? Who is in your
Bubble.
Zoom bubble.
bubble? Any other phrases for it?
Um,
No.
I don't
This
know.
was new to me too. I like this one. Quarantine.
Oh yeah,
Quarantine.
I got my husband a shirt for Father's Day that had quarantine on it from my
Quarantine.
daughter because they're quarantining at home together. So I should have thought about that.
All
That is
right.
like, that's got Etsy written all over it. Quarantine.
Quarantine. It does.
I
It
think
does.
because Life is Good had them. Yeah,
Nice.
that's cute.
All right. This next one is kind of pedestrian. I'm going to skip it. It was coronation for a vacation that you take at home. What do we call someone who is annoying because they will not wear a mask?
Oh, I could think of many words for
Yeah,
that.
those are along with the words that I can't
This is the one that's closest to being not appropriate for a family podcast. Are we a family podcast?
Ooh, Alex spicing it up.
I know. I'm scared to even guess because I feel like I might be censored in the guessing process.
We don't. What's the question again?
Someone who's annoying. They will not wear a mask.
Well, I don't know. I just think of them as anti-maskers instead of maskers.
Any shots, Trish? What do you think?
No, I know. I'm
wait,
not thinking of anything clever.
COVID.
All right. Covidiot? Yep. Covidiot is one of the 2 that I have written down. The other
Oh,
one is
that one's good.
the other one is mask hole.
Ah, that's good.
That is great.
Using that.
I'm definitely using that too.
Well done. I think you got most of them. Nice work, everyone. Thank you both for joining me. This has been a lot of fun. I'm going to sort of skip my usual postlude here because we are at an hour and 10. But again, Trish, Melissa, thank you both. Appreciate it very much. This has been great.
Yeah, thank you, Alex. It was a pleasure.
Yeah,
Great
thanks
to
for
see you.
having me.